Good morning. I hope you are refreshed now, and it is a fourth day, and I hope you are enjoying the journey. And we came across so many topics related with the in vitro as well as in vivo studies, and starting from the natural compounds as well as the mechanism of the action of the molecule, and in future also we are going to study so many other aspects. And whenever you are studying all these molecules or basic study, if you have to do, we are using the cells for our studies, and that's usually we go for cell culture techniques. In this tutorial we are having few questions, and then we will I sent you already the study material today morning, and the question start with what is actually the cell culture. Cell culture is basically it is growing the cells maybe out of the living organism. You are taking out the cells, and in a controlled atmosphere you will maintain the pH, you maintain the conditions which are existing in our body to we mimic the same condition outside in the laboratory. That we call it as tissue culture or cell culture. Actually it started in long back, and that time when it was started, actually the scientists, they were growing in the beginning these frog embryos and all. It was they were using those the cells majorly because these cells or the organisms are actually the cold blooded animals, so you need not to provide them the the incubation or temperature and all. So they were thinking like that, and later on the study went on, and they started making use of different media. Earlier they used to take some extracts of the living organism, and in that one they used to place the tissues, because initially this study was majorly related with the tissues, and most of the occasion overlappingly we use the terminology tissue culture and the cell culture. But that tissue culture, whatever it was done long back, the same terminology we are using now also. But the way we are doing the cell culture, from maybe compared to earlier days, it is totally different. We have a definite media, we have a definite protocol, we do have so many aspects that has been changed. And the other question was, what is the good laboratory practice, one thing, and what are the minimum requirements for a cell culture laboratory? Because being the students of this, maybe the cell culture or any other technique, first of all, when we go to the laboratory or if we are designing the experiment, we should know what do we require. That is the basic thing. And next thing is that what we should do and what we should not do. That we consider it as the good laboratory practice. Because there are certain things that should not be done in the laboratory, and there are certain protocols that should be followed must. That we call it as good laboratory practice. And here minimum requirements you need, as you know, when you go to the cell culture lab, you come across with, and if you see any laboratory, you find that one thing that is common in all the cell culture lab is biosafety cabinets. That is actually the place where you are doing the handling the sterile material, including the cells. And along with that one, you need even cell culture media. So in order to maintain the cells, you need certain media or the growth, maybe the one which supports the growth of the cells. If you are not providing the nutrients properly to the cells, they may not able to grow properly. So we have to discuss about the different media. And another one is that regularly we are using phosphate buffer saline, we will see later. And what happens is that there are certain cells they actually adhere to the surface of the flask or surface, because once they attach to the surface, we are supposed to remove them, because if they grow in number, which is if they reach certain number of cells, we are supposed to subculture them, just like what you do in microbiology. You are continuously maintaining the culture for long time by subculturing. Here we are also subculturing this the animal cells by using because we have to detach if it is an adherent cell which is attached to the supporting material. Then we are using trypsin EDTA, and later we need even centrifuge and centrifuge tubes, because sometimes you need to centrifuge and concentrate the cells if you have to go for further seeding. And later on, whenever you are doing certain experiments, you need to know how many cells you have seeded in the tube. In such condition, you should have a knowledge about how many cells are present in that medium. So in order to do that one, better we should have hemocytometer, or sometimes if the lab is well funded, the better you can go for the automated counters. And even we need flasks, maybe for seeding the cells. And what should be the laboratory setup, because along with that one I can tell what and all that is needed for the basic cell culture lab. Here we need the dedicated space, especially for doing the sterile work, where you are using the separate chamber or separate room for handling the cells. Here what we will do, we have a lab, suppose say for example if I have this room and I cannot use completely, because if my space is limited, I cannot use everything for the cell culture. Better thing is that you make a partition so that the you can avoid so many contamination in that area. So by partitioning that area from the other area, because you are not only doing the cell culture, while doing the cell culture also you are doing various other works. You are supposed to prepare the media. Suppose if you have not purchased the prepared media, you are supposed to prepare the media, or you may have to wash the glasswares, or you may have to keep other chemicals, or you may have to keep the antibodies, or you may have to store the media and all. So for that and all you need a space. Everything cannot be clumped at one region in the cell culture laboratory. So what you have to do, better you keep the space separate for the cell culture, and that area let it be separate from all other activities which you are doing. And sometimes maybe you make a partition in such a way that whenever those who are having the work in that area only will be there in that particular area. Otherwise what happens is that there is a chance of the contamination, because you are going from outside, and there is a chance of taking with us, maybe there are contaminants, and that may affect your the studies. So you have to be careful about that one. And one more thing is that you need the controlled environment. You are supposed to maintain the temperature so that the media is not disturbed, or even the cells are not disturbed, and you may have to use a proper environment. And work zone, what way you maintain the work zone? You need a clean area, that is for storage of your media and the reagents or equipments, and where exactly you are doing your culture work, that is biosafety cabinet, it should be very clean. So that because why I am telling all those things, because you think that you have done a very good job, but you don't know what are the contaminants you are introducing when you are doing the work. Because there are certain cells, even if you are handling, they may actually cross contaminate the other cells. This is what happens especially with HeLa cell lines. So this was actually one of the oldest cell lines, that is immortal cell lines, that is maintained. It was in 1951, it was taken from Henrietta Lacks, a cervical cancer patient in John Hopkins University, and it is growing, and the number of cells that are used in the lab is more than her size itself. So when she died, she was around 31 years age, and nearly 50 to 70 years, so many this cells, or so many times we have used the cells, and different cell suppliers, they are giving the cells. We maybe many people they study or use that one for various cellular maybe cell related activities. The major problem with this HeLa cell line, because I am taking one example and it will be easy to understand. These HeLa cell lines, even they can be, they can cross contaminate and dominate other cells, and they can grow in that one, and they prevent the growth of the cells which you are interested. And they can even actually transmit through the air also. That's what some studies says that. So in such a case, and suppose if that is the case, if there is a one aggressive cell, and which even if it gets transmitted or contaminates through the air droplets, then your study is gone. You will not know whether you are growing the HeLa cell line or you are growing some other cell line. So in that case, if you don't maintain your culture area properly, then there will be problem. That's why we need to maintain always this culture area that should be properly maintained, and it should be neat. And once you got a clean area, you need to maybe incubate the cells. Cells, they have to grow in the media. So if they have to grow in the media, we are supposed to give them proper temperature and the conditions which favors the growth of the cells. Therefore, that one we need the incubator, and at the same time you need even waste disposal area, because whatever I am telling, you can observe in the cell culture lab whether we do have that kind of facility. Because usually when we go, if we don't have the knowledge about that one, that's why we kept this tutorial first, so that you are sensitized to the things what it is done at the cell culture lab, and you can see what and all the way that has been maintained, and why they have certain restriction when you are entering into the cell culture lab. So along with that, as I mentioned, let me tell about the biosafety cabinet also. So biosafety cabinets, actually it is necessary for aseptic handling of the cells, because you don't want anything to contaminate your cells, and you are running a costly show. By chance, if there is a small contamination, and if you are not able to predict whether it is right or wrong, it may affect your results, and you will not have the reproducibility of the results. So for that one, we need type 2 A2 biosafety cabinet, because that is considered to be the one which regularly we are using for the cell culture studies. And not only that one, so one thing is that we need a separate area. Second thing is that we need the biosafety cabinets, we need the incubator, and we need the inverted microscope where we have to observe these cells. So cells can be observed, or morphology of the cells can be observed using these inverted microscope, because the cells contain the media. You cannot take the normal microscope, usually what we are using, that is upside down. It may not give you the clear picture of the cell. So suppose if you are observing from below, you will get a clear picture of that cells. That is what actually inverted microscope is. So that we will use. And sometimes you have to, because you have to seed the cell in the sense you take a stock of the cells, and or if you get cell from cell repositories like NCCS Pune, you are supposed to store them or you are supposed to seed them. Seeding in the sense you are growing them. When you have stored that cells for long time at minus 196 degrees Celsius, that is the liquid nitrogen can, so it is at maybe actually it is straight away you cannot use the culture, or you have to thaw that one. So it should come to the kind of the 37 degrees Celsius. You immediately thaw those the vials or ampules at 37 degrees Celsius, and after that you will seed into the flask that contains the media. And there you need the water bath to maintain uniform temperature, or even autoclaves, because if you have to sterilize anything you need the autoclaves, and refrigerators and freezers. And there are different kinds of refrigerators maybe that are maintained at 4 degrees Celsius, and there are the one which are maintained at minus 20, and there are certain freezers, they are maintained at minus 80. All these things we are using for storing different the chemicals or solutions based on the necessity or based on the conditions. And as I told, liquid nitrogen is used for storing the cells. Usually whenever you are storing the cells for the long time, you need something which prevents the distortion or maybe the deformity, or even the spoilage of the cells if you are storing for a long time. So in that condition, maybe that case you can go for the very low temperature, that is minus 196 degrees Celsius, and along with that even you need pH meter. Suppose if you are setting the pH, usually nowadays nobody goes for the preparation of the media. Earlier days they used to take the powders, and that powder used to be dissolved in the big container or small container, whatever you need, and it used to be filtered, and then it used to be used. It was used to be used. But the thing is that there is a lot of chance of contamination, and nowadays as many prepared medias from the suppliers they are available, instead of working so much on those wasting time on the preparation of the media, you can straight away purchase that the prepared media which is already it is present in a aseptic condition, and you can use that one. It will be much easier. Or if you have the facility to prepare the media, that is fine. Or even osmolarity can be checked. Suppose if you have osmometer, and these are all actually the basic materials that are required or equipments. And along with that even we need certain consumables as well as the accessories for our studies. So we need tea flasks, and there are different sized tea flasks they are available to maintain the adherent cell. Here those who are not familiar, just I mentioned what are adherent cell and what are suspension cell. So adherent cell is the one, if you take the cells and if you grow in a proper surface, they come and bind to the surface and they fix to the surface, we call them as adherent cell. And those which are actually present in the suspension, they won't come and bind to the surface, solid surface, we call them as suspension culture. Okay, those who are studying biochemistry or maybe others, they are familiar, but there are some people from the chemistry, they may not be familiar, that's why I am telling about that one. And you need even petri dishes, maybe if you are growing the suspension cultures or maybe any studies if you have to do, there are different petri dishes we are using. So there are different sized petri dishes are also available, like 35 mm or 50 mm or even 90 mm, depending, or even bigger than that one that are there. Usually we go up to 90 mm when you are going for the cell culture, but rarely even the bigger one they are used. And multi-well plates, so usually either it is a 4 well plate, sorry 6 well plates, or even 24 or 96 well plates can be used, or as Dr. Nag was telling, even higher number of that the wells they are available, so that can be taken, multi-well plates. And spinner flask, that is majorly used for suspension cultures. And whenever we are using the or handling the cells, usually we go for the pipetters. Earlier days, usually they used to use serological pipettes. Whenever I was doing the PhD, I used to use that serological pipettes, and we used to keep on a bulb, and then you have to transfer that media and cell using that serological pipettes. But nowadays, the automated pipettes, they are available, and if you are using these microplates, that is containing 96 well plates, even multi-channel pipettes, they are available, and even the automated pipetters, they are available, so that depending on your fund availability, you can plan and you can study, or you can make use of this one. And even the micro pipettes, suppose if you have to add say for example very low quantity, so you can use that one. So serological pipettes usually we use for 1 ml or 5 ml, 10 ml, even 25 ml we use serological pipettes. But if the volume is less, the maybe there are pipettes, they are available, even that micro pipette we say, or pipetteman. So even for 25 ml we do have, 5 ml we do have, or even the smaller volumes like 0.1 microliter, all those things. Depending on the necessity, you should plan what type of this the pipettes you are supposed to use. And then what is the other essential commodity, or maybe the material that is required for the cell culture lab? The major thing is that we do need the media, different medias that are regularly used for growing the cells. The major medias that include are basic medias like DMEM, Dulbecus modified essential medium, or RPMI, or minimum essential mediums. And along with that, even we do use the FBS, or fetal bovine serum, or sometimes even serum free media is used. So because sometimes maybe that depends on the cell, that's what I am telling. It depends on what study you are going to do. Accordingly you should select the media, and accordingly you should select the flask, or accordingly your maybe the other materials that are used. And we need to remove the cells or detach the cells from the attached surface. We are using trypsin EDTA, phosphate buffer saline usually we are using for washing the cells, because sometimes if the cells are to be detached, and if you are adding the trypsin, and if you are not properly washing that the cells, usually trypsin may not act, because the serum might contain trypsin inhibitors. And sometimes we need to add even antibiotics, because we want to take care of the cells that should not be contaminated from the microorganisms. So in order to do that one, what we will do in the media, we will add antifungal and antibacterial the chemicals like penicillin and streptomycin combination we will use, so that we can avoid the most of the microbial contamination. But there are certain contamination, they go beyond even the microbial contamination, that we will see later. And even plastic waste, whatever we need, or even glass bottles, media storage. And I am actually clubbing these two materials and the good laboratory practices. Okay. So whenever you go to the lab, you should know what you are doing and why you are doing. And maybe this, if you are going to the cell culture lab, better is that you wear a lab coat, so that you are not taking any contaminants from outside. Okay. cells are sensitive to take up that contaminants, better you avoid all those things by the dedicated lab coats so that you can avoid most of the contaminations. So usually the country like us, we have a dust problem. Maybe that dust may affect your culture. So better you take care of that one. And once you are handling the or once you are working in the hood or biosafety cabinet, better wear that one. Or suppose if you have to talk, or better you should not talk when you are working in the this biosafety cabinet. Make sure that it is covered with a sheet so that you will not spill anything on the media you are working. And make sure that UV sterilization of that area, because that is the best way you can avoid most of the contaminations. So here even the biosafety cabinets, they come with UV sterilization. Best way is that you swab that the biosafety cabinet before work and after the work. And before work you switch on the UV maybe for 15 to 20 minutes and switch on the UV after the work also for 15 to 20 minutes. So that is the good way you can avoid most of the contaminations. And moreover, you are making a way for the other person to work on that. Maybe whenever too many people they are working, there is a chance of crowding. So that may affect your work. So try to make your schedule accordingly. So whenever you are having too many people, you are having work, you fix the time. So that if you have the work, no one else will be, maybe they need not to expect you to finish off faster or something like that. You take your own time and finish off the work. And when you are doing something hurriedly, there is a chance of making mistakes. So you avoid such things by making sure that you are having a proper time, you schedule a proper time, and then you work. And decontamination protocols. Not only working is important, after that how you decontaminate the things what you have used. Regularly we use, suppose if you have to work in the biosafety cabinet or any culture work, better you swab your hands with the 70% alcohol. So that avoids most of your the contamination from your side. Okay, it has been shown that even the cells that are dead cells present on us, it may transmit it if you use the bare hand. Suppose if you are wearing the gloves, better you wipe once with even 70% alcohol. You can avoid, maybe by chance, if there is any chance, you are avoiding. But ultimately, the thing is that you should avoid the contamination. And biohazard material should be thrown or maybe disposed at a proper place. They should be designated with biohazard symbols. And most important thing is that whatever you do, you keep the record. So unless you keep the record, you don't know what you are doing. Suppose you go to the cell culture lab, you did something and you don't know what you did on that day. Next day you will be repeating the same thing. You did not know what you have done in the previous day. So then it is a repetition of the work. And moreover, you will not recognize what are the mistakes you are doing when you are working with the cells. So try to figure out what actually you are doing and what are the challenges you are facing when you are, because initially when you are starting the cell culture, you are new to the technique, you are new to the work. When you are new to the work, always make sure that you are maybe following a proper protocols so that even others they can work smoothly in the lab. You maintain a proper lab notebook, that is one of the major aspect, and you maintain inventory management systems. You maintain how much consumables you have, how much stocks you have, so that your planning the experiment will be easier. And you maintain certain standard operating procedures. So that SOP will help you move in a proper direction. And the other question was that what is the role of carbon dioxide incubator in cell culture? So here we usually use carbon dioxide incubator for most of the cells. So carbon dioxide incubators, earlier they used to provide the temperature and carbon dioxide that used to be supplied in that chamber. It maintains the humidified condition so that the media will not get dried. Okay. So then here nowadays even the chambers that are available, they are coming up with various adjustments, like you can sterilize that the carbon dioxide incubator by increasing the temperature inside that the chamber. Earlier you were supposed to the cleaning and maintaining the sterilization was a big challenge. Now even the suppliers, they are designing in such a way that they are built in with certain kinds of sterilization. Suppose you can take the temperature up to 180 degrees Celsius and you can make sure that your carbon dioxide incubator is free from the any contaminants. Okay. And at the same time, whenever you are using the carbon dioxide incubator, usually you fix the carbon dioxide around 5%, and carbon dioxide present in the chamber, it diffuses into the media, and it may interact with the media to form that, the reactions. Those who have studied biology, it becomes carbonic acid, and it becomes bicarbonate and H plus. So it will help in the proper maintenance of the medium. So whenever you are using the carbon dioxide incubator, there are certain precautions you need to take care of. The major thing is that because all of us, we will be keeping in the same incubator, we cannot have 10 incubator at a time. So whenever you are keeping certain cells for incubation or any maybe majorly the cells, make sure that the whole the area inside is clean, and regularly make sure that area is kept clean always. You wipe with some the alcohol, or you sterilize that one with the high temperature provided in the incubator. Make sure that that works fine for others also, because when too many people are using, naturally the chances of the contamination will be a bit higher. And sometime if you forget to close the cap of the flask, that it may spill something and it may contaminate others' work or even your own work. That you have to be careful. And the other thing was that I already told what are actually adherent and suspension cultures. Adherent is the one where it is attached to the surface, and the suspension is the one which is not attached to the surface. So why fetal bovine serum is commonly used in cell culture media? So this is actually there is so many studies that are going on. So fetal serum, this fetal bovine serum is actually a costly one. So sometimes you may have to add it, or sometimes by adding itself you are creating the problem. That's why here fetal bovine serum, it is rich with various nutrients that are necessary for the cells to grow. Okay. So if you provide, maybe there are certain media you are supposed to provide that fetal bovine serum for the growth, like DME medium, you have to provide 10% FBS, or for that matter you may have to use serum regularly for the proper growth of the cell. It depends on the type of the cell what you are using, and type of the media what you are using. Because the major function of the serum is that serum will contain the nutrients, and that nutrients may favor the growth of the cells, but it should not hold good for all. Some cells may not grow properly when you supplement with the FBS. In such occasion, there are certain serum free medias available, and you can go ahead with those kinds of serum free media. Or here majorly sometimes there are some disadvantages of the FBS also, because you cannot maintain the batches, this homogeneity of the batches. You get the serum from the supplier in one batch, and the same batch might not be supplied by the supplier next time. So you cannot maintain or control the experiment properly. That's why you have to make sure that whenever you are using this serum, you should aware that whether it is giving the proper uniform growth of the cells. And at the same time, sometimes even the presence of the serum also affects your studies. That's what I was telling previously. Whenever you are actually going for the detaching the cells, or we call that word as passaging, where you are subculturing, in that time, where suppose if there is a small quantity of trypsin, it will actually, a small quantity of serum, it prevents the action of the trypsin. The best way is that you wash the cells properly before going for that one. And how does an incubator help maintain the optimal cell growth conditions? That's what I was telling. The temperature, this incubator provides, because this carbon dioxide incubator and the incubator I am going in line with line, where the majorly we are using carbon dioxide incubator while growing the cell, but in certain conditions you may have to have maybe the normally what you are using the incubator without any carbon dioxide supply, where you are setting certain temperature and you want to set the reactions, and that can be done there, and that's where you will get help from this incubator. And how what are the key aseptic techniques required to prevent contamination in cell culture? What are the aseptic techniques? In the sense, that's what actually the good laboratory practice, what I am telling. So when you enter the lab, make sure that you are wearing the lab coat. And once you enter the lab, make sure that you are tying your hair. Okay? Because once you are working in the biosafety cabinet, so that may lead to the contamination of the cells. Okay? Or along with that one, regularly swab your hands with the alcohol, and sometimes what happens, you take the media, and that media you take it into the biosafety cabinet for doing the work. You have taken the media, inside the media it is actually sterile, but the surface may not be sterile. So that will be the source of the contamination. So what do? You properly wipe the culture bottle properly with 70% alcohol, and make sure that keeping in the biosafety cabinet. Okay. And at the same time, whenever you are in the biosafety cabinet, make sure that you are not talking, and your hand should be at least inside about 4 inches inside, so that it will be properly inside that biosafety cabinet, and you are working, and you are not disturbing the movement of the air. And earlier days, the biosafety cabinets used to be provided with the burners. Usually whenever we were transferring the media, the transferring used to be carried in presence of the flames. But nowadays the biosafety cabinets are fitted in such a way that usually we avoid the burners, and you should have a proper arrangement of the materials what you are using. When you go to the biosafety cabinet, you keep the things what is required. Sometimes what happens, we have the habit of thinking that we think that we assume that we need so many things and we keep everything piled up in the biosafety cabinet and you will not able to work. And sometimes whenever you are using the pipettes, the major mistakes what we do, we touch here and there. That may get the contamination. Or even make sure that whenever you are sitting also, you are sitting in a proper posture, and at the same time you make sure that whenever there are certain biosafety cabinets, they are coming up with certain sheets so that your breath will not reach the incubator. That you have to make sure. And whenever you are handling any instrument or any flask, because media you have to transfer. Best way to transfer the media is that either you take the dispenser, either you take some pipettes or you transfer it. Suppose if you have to transfer the media using the bottles only, best way is that after transferring, better you wipe that the bottle properly. Otherwise what happens, that it spills around the rim of the bottle, and that itself, it is a contamination. Okay, so that care you have to take. And so one thing is that another one, what is confluency and why should cell be passage 70 to 80% confluency. Confluency in the cells, when you grow the cells on the culture flask, they will actually maybe they grow, and if they grow 100%, they may start getting detached. Okay, so that may not give you a proper results when you are going for this subculturing, or we call it as passaging. So what best way is that you take the culture media with the cells, you observe under inverted microscope, and then make sure that it reaches only around 70 to 80%. So once it reaches that maybe the stage, then immediately you go for passaging, that is subculturing of the cell. And by that way, what you are doing is that you are avoiding the losing of the cells. Cells, they will start getting detached if the number of the cells, they become increased. And what steps are involved in subculturing or passaging cells? And here, when you are passaging or subculturing the cells, subculturing the cells, what you have to do is that, so what you will do, you will first take the culture and you keep all the things and you follow the precautions whatever I was telling, and then you take out the culture flask in which the cells are growing, and you keep on the this, and make sure that whenever you are doing such things, you keep the laminar airflow. It should be free from whatever that is not required. Make sure that you are keeping that maybe trypsin solution or media whatever you required, or the pipettes whatever you require, everything keep it ready. And then you take the cells and then decant that media whatever that is already present in that culture flask, and wash those cells with the PBS, so that if you wash that one, it is easy to treat with the trypsin. So you add the trypsin, and when you take the culture flask, and if you just add it, there is a chance that only certain regions they are reacting with the trypsin. Make sure that the whole area is covered with the trypsin, even if you are adding around maybe 0.25 ml or whatever may be, depending on the culture flask size, and then make sure that it is completely covered that cells, and you wait till the cells they start getting detached, and then you add the media, because here the advantage is that if the media contains the serum can, this FBS itself can act as a stopping agent for the trypsin. So once the cells are detached, you think that the cells are detached, then you add this FBS and block this the further reactions, and you make the suspension of that the cells, and then you have to dilute it. So dilution in the sense, maybe you have got one flask of cell, and that one flask of cell should be divided either 1:2 or 1:3, 1:4, depending on the type of the cell and type of its multiplication capacity. So if the cells are dividing very fast, and if you are giving only 1:2 dilution, it will grow very much faster, and you will not have much time to, you should only focus on passaging the cells, no other work will be left out. And once you do that one, and before doing that, you should know how much cells are present in that the suspended one. You take out and find out how many cells are there. Accordingly, you find out how many the culture tubes in which you have to seed the cell, and then you once you subculture them, keep it in the CO2 incubator, and that day work is done, and clean your that area properly. Make sure that you switch on the UV light for 15 to 20 minutes, and you keep it at in the CO2 incubator, and next day morning you have to come and observe what is happening, because if there is any problem, cells they may not attach to the surface, or there may be some problems, or sometimes the cells may not be detached properly. You find out what is happening in that time, and accordingly you have to design. Sometimes what happens, the cells will not detach only, and that time you have to increase the trypsin concentration, or sometimes the cells will not grow only, maybe because you have added too much amount of trypsin, or you have kept it for long time for the action of the trypsin. So once you can analyze after the next day, once you come, you will get an idea whether you have done it properly or not. This is what you can do. Other things you should be aware that whenever you are doing is that contaminations which you cannot able to recognize, or there are contaminations which you are able to recognize. The contaminations from the bacteria and fungi, it is very easy to recognize, because immediately what happens, the media is added with the phenol red. That phenol red is having a color, and as the pH reduces, that color becomes actually yellow. If you are getting yellow color or turbidity in the media, immediately you can suspect that something happened in that culture. So immediately you better check for the turbidity as well as the color of the media. So, or sometimes there are certain contaminations, it is very difficult to detect. That's why when you are purchasing the cells itself, you should be very much careful. The major problem with the contamination is that mycoplasma contamination. They are the bacterial type of cells without the membrane, and they grow in the media, and they are not acted upon by even the antibiotics. And actually they contaminate almost all the maybe the cells once it is infected in the lab. And moreover, they cannot be visualized under microscope also. So there are different methods that are available. By that one only we can able to detect their presence. Either we have to go from culturing the mycoplasma, or you have to go for 16S rRNA sequencing, whereby you can find out the presence of the mycoplasma. But the thing is that they don't affect too much your the cell culture. You will not able to recognize, but the result what you are supposed to get may get altered because of those organisms. That's why you have to be very careful about the mycoplasma contamination, and that's why the best way is that you make sure that when you purchase the cells, always make sure that you are purchasing from a proper supplier. So authentic suppliers, those who maintain the kind of the American type of culture system discipline or something like that, they mention we are following this method to maintain the cell culture. That would be the best, because here our regular source for the cell culture is the NCCS Pune. So they are maintaining many cells that are regularly used for our studies. So those can be used, and you are sure that they have done all the studies. And if you want, if you suspect there is problem with the contamination of mycoplasma or something, best way is that you can send the sample, because if you don't have the facility, better you send the sample to NCCS only, get it checked, and they do the procedure and they will find out, they will tell you whether you have done is proper or not. And at the same time, always try to avoid, that is the best way. The prevention is better than cure, isn't it? In that way, we have to avoid the contamination. And in between, I did not mention one thing. So usually we do the cell culture, either primary cell culture or the cell lines. Primary cultures are actually, it is straight away derived from the tissues and we grow them. Whereas cell lines are the one, they continuously maintain that line of the cells, that maybe when we are in the laboratory you can see the cell lines. We are not maintaining the primary cultures. Usually whenever we are studying, because in Nag's lab they used to do even that one, because pancreas tissue they used to take and they used to grow also, I have seen that one. If you are working with the cell culture, here we can work, or even you can do that one also. If your work, if it demands, you can do that one also. So that's about the tutorial, and if you have any other doubts also, we can discuss. So as we are running out of time, still if you have any time, doubts, we can discuss at the lunch time and free time.